Okay, let's just go back to recap from last time. We did the overview of monohybrid crosses using the letters, and then extending that to multiple alleles where gene I should have been for the ABO blood group, and it comes in these three varieties: IA, IB, and IO. IA and IB being codominant, IO being recessive. And our various combinations there, of course, which gave me my final IO. And I didn't show you any crosses with that, but it might be as well to do that. Would it be possible, for example, for somebody with group A and somebody with group B to have a child with group O? Yes, it would. Yes. Because the person with group A can be that. The person with group B can be that. Now, combinations from those two parents could be, I'll just do the groups. If we put together that one and that one, we get AB. If we put together that one and that one, we get A. If we put together that one and that one, we get B. And if we put together that and that, of course, we get, So you can actually get all four blood groups if you've got parents who are A and B. But that's only if their alleles are IA-IO and IB-IO. If you've got IA-IA and IB-IB, then yes, you shouldn't be getting any children of blood group O. And in fact, that wouldn't even just cast doubt on the father. It would cast doubt on the mother as well, wouldn't it? So, yeah, that's not going to happen. So the multiple alleles thing, you've got to remember, you can have all sorts of combinations. And we've got a lot of other characteristics that are controlled by multiple alleles. And that can actually, create all sorts of possible combinations. The other thing we looked at was the idea of lethal alleles. Or another thing we looked at was lethal alleles, where you could, for example, have a genotype or a phenotype, I should say, just not showing. Reason being that a particular condition, a particular genotype just stopped development. Now, it could be before birth, it might be that those embryos never develop, or it could be life-limiting in childhood, say, so that you never get that particular genotype maturing to reproductive age, and that it can distort the number of individuals in a population that you would expect to have certain characteristics. Now, this is really important as we go on to population genetics, because you can actually make predictions about the percentages of individuals in populations who have certain genotypes or who have certain alleles. But if you've got a genotype that's lost either before birth or in childhood, and you're looking at the genotypes of the adult population, the breeding population, if you like, it can completely distort it. So worth bearing that one in mind. I used Manx cats as an example for this one because it's a fairly well understood one, where Manx cats are the ones with no tail, but they are heterozygous. Because if you get the homozygous situation where the spine doesn't develop, then the embryo is lost. And then you can have, other examples we will leave. Oh yes, we then went through all the sex-based inheritance, didn't we? So this is actually really complex. And if you remember, it's only one small part of the Y chromosome that's actually important. The SRY gene is the only bit that actually matters in terms of switching on male development. And even then, the body has to respond to the testosterone in the mother's body. And so basic inheritance like that, but then other sex-linked disorders have to be noted on the chromosomes themselves. So used colour blindness as an example. And then we looked at this idea of X inactivation, which I find an absolutely fascinating subject. That you get this switching off of an X chromosome in every female cell because having two X's is a bit too much. So you get the one switched off condensing into the Barr body, and in most women that will be about 50-50 distributed evenly over the body, but it doesn't have to be. Chance is such that you could have all the same chromosomes switch off, or it could be sort of all of one of them switches off on one side of the body and all of another one switches off on the other side. You can have whatever combination is possible. And, even if it's as unlikely as winning the lottery, it can still happen, can't it? And, the best example with this one, of course, is the tortoiseshell cats, which, I think is a really nice one. You don't get tortoiseshell males because the only way you can get tortoiseshell is the co-dominance between XB and XG. And if one switches off in every cell, then the other one is functioning, so you either get the cell giving rise to black hairs or ginger hairs, or orange we should have said, and of course you don't get that in males because you've only got one X chromosome, so we only got the black and ginger possibilities. So that was as far as we got, wasn't it? I think, yeah. Okay, the next task is to have a look at the dihybrid crosses. So I want to do that, and then I think we need to start looking at some of the questions in the folder because we haven't been doing that. So we'll have a look at that, and I'm toying with the idea of recapping something that we did in the first biology unit, and that was when we were doing the statistical bit, and we mentioned the chi-squared test. Chi-squared test, don't pull a face. Chi-squared test is so relevant to testing dihybrid ratios that I think we sort of have to. We may not sort of, I'm not going to find you a whole load of examples to work out or anything, but I do think I need to go back to it and just explain, because at the time we didn't really have many examples to apply it to. So I think that one's got to return to us. All right, dihybrid crosses then. Now, the situation here, we have to make it clear that when we're looking at this, it's the inheritance of two characteristics, but they have to be on separate chromosomes. If they're on the same chromosome, then they may not be separated when gametes are formed. If you remember that bit that I keep saying I need to do with the other group, the bit about how meiosis contributes to variation. One of the things in there is crossing over. Well, you can imagine if you've got two characteristics on the same chromosome, if they were next to each other, then they might travel together for a start. The second contribution to variation is the fact that we produce haploid gametes that then produce new combinations at fertilization. And the third thing is this independent assortment. But if you've got two characteristics on the same chromosome, those characteristics are not going to be independently assorted. They're not going to move independently of each other. They will move together. So the assumption that we make here when we're sort of modelling a dihybrid cross, as it were, is that the characteristics, the genes, are on two separate chromosomes. Now, this is why it's quite important to bring in the chi-squared test, because what we end up with for the this particular type of cross is a ratio where we had Mendelian ratio for the monohybrid cross. We're going to end up with the dihybrid ratio for this one, and you can then test against that as if we are expecting the dihybrid ratio in our offspring, but we get some result that we can't really decide whether it's close enough to the dihybrid ratio or not, you can use the chi-squared test, and if it's way off, then it might be that those two characteristics were on the same chromosome, and you can claim possible linkage between them. So although that's not something I'm going to put in the exam, it's an obvious sort of application of it. So I do need to do it, but I will try and make it as painless as possible. All right, so this is inheritance of two characteristics whose genes, that's not good English, should put the genes for which are on separate chromosomes. Right, because you then don't get any interference by processes like crossing over and the assortment process that's going on. So the example that we always use for this is Mendel's peas again. And Mendel's peas always looks odd, doesn't it? We're going to look at colour like we did before, but now shape, the shape of the pea. The pea, of course, is a seed, so we've got the pea colour. So we've got the big Y representing the dominant yellow, and the little y representing green. And then we could choose anything. You know, it could be the height of the plant, the colour of the flowers, all sorts of things, but we just tend to use this one, pea shape. And the dominant shape for peas is round. It makes you wonder what the other shape is. It is wrinkled, yeah. But, you know, don't you wish it was sort of, I don't know, cube or tetrahedron or something like that, you know. You'd find one of those occasionally, that'd just be so much more entertaining. Good way of getting children to eat vegetables. Right, so it's a little r. It's actually for wrinkled. Now, the process that Mendel went through was the same as for the monohybrid cross, taking pure breeding individuals, and this time you'd have to follow them over generation after generation to find out, well, you had to do the monohybrid cross first to find out which the stronger characteristics were in either of these situations. Trying to do it without mentioning the word gene. When I talk about Mendel, because Mendel knew nothing about genes, I always try, if I can, not to talk about dominant and recessive alleles or genes when actually talking about what he did, because, you know, he wasn't working on that principle, was he? You know, it wasn't until much later that, you know, all that was worked out. But his first cross involved two parent plants which were pure breeding for either the dominant characteristics or the recessive. So the first cross was pure breeding, which of course we describe as homozygous. Right, round yellow. I don't really need to do that with a capital R, to be honest. That's going to confuse matters. Round yellow crossed with wrinkled green. Right, now you should be able to tell me, I would think, what the offspring were going to come out like. Yeah, they're all going to be round yellow, aren't they? Because that's the only option. We know that the only allele this parent can contribute to every offspring is a dominant R and a dominant Y, they're all going to come out round yellow. But if we do this, putting all the details in, these would be the parent phenotypes. We then have our parent genotypes. So the round yellow, what's the genotype of that going to be? Big, yeah, big. Right, big R, big R, big Y, big Y. I'll put them in that order because, as I said last week, we do them in alphabetical order like in algebra. You know, it doesn't matter what letters you choose. So sometimes you might find that they go the other way around. If you'd, yeah, I don't know what you could choose for C for colour, perhaps, rather than Y for yellow. But oh, and of course the wrinkled green will be, yeah, little r, little r, little y, little y. Okay. The next thing to consider then is the gametes that we can get. So the gamete genotypes. Now, when it comes to exam questions, this is where people go wrong. Because they will take this one, and I don't know what happens to people in exams, but it's, it's always the same, isn't it? There's always something that afterwards you think, did I really write that? What gamete genotypes can we get from the round yellow? That's all we can get. But when it comes to exams, people will put big R is one of them. Big Y is another one of them. And I think to myself, why are you writing that? And the other one, of course, yeah, is the little r and the little y. And you've got to remember that a gamete contains one copy of every gene. So, in a human being, where you've got something like, I don't know, 20,000 genes, then if you were to write out the gamete genotype for a human being, each egg, each sperm would have to have 20,000 of those. All right, 20,000 letters, wouldn't you? All right, giving you the, the status of every single gene. So a gamete contains one copy of each one. So our combinations here are really not terribly tricky to do. So I'm going to miss out the crisscross because you can't really do it with this anyway. It doesn't actually work. So we're going to say, I'm going to call them F1 genotypes. So the only genotype we can get is that. And that makes sense, doesn't it? The offspring can only inherit one dominant and one recessive allele for each characteristic. Written in alphabetical order, uppercase before lowercase, that's the convention, just to make sure that if we get it put together in some other way, if it was to look like little y, big R, little r, it still means the same thing. So, you just gather them together in a particular order. That is, it's not, doesn't have any different meaning, it's just tidiness and convention. So, we know the F1 phenotypes then. Just as in the monohybrid cross, they are all, so all round yellow. Okay, so far so good. Fairly straightforward. The second cross takes two F1s and interbreeds them. Self-pollinates them, if you like. So that you're mixing the same genotype. So it's going to be big R, little r crossed with big Y, little y. Obviously, this gets a little bit more complicated. Oh, what did I put up here? Did I put first cross? Yeah, first cross, right. Second cross then is two F1 individuals. All right, so they are both heterozygous for both characteristics. Okay, now we can get a little way through this before it starts to get a bit tricky. If we start off, let's head off the same as we did before. The parent phenotypes, we know this time they will both appear to be round yellows, won't they? So round yellow. But we don't actually know just by saying that what the genotypes are. We do, in fact, know because of how we produced them. So the parent genotypes are what we ended up with at the end of that first one. So big R, big Y, little y. Now, this is where it then does get complicated. No, it doesn't have to. But we've now got to sort out our gamete genotypes. Now, we've got the same parent genotype for each one, so it's going to be the same for both of them. But what are our possible gamete genotypes for this one on the left? How many are there? How many possible gametes could you get out of that? 4, yeah. So we've got, give me one of them. You would choose that one to start with. I want the I tend to go, there's a sort of order for this. You'd have the big ones together first. That's usually how you'd write it. Then you can have big R, little y. Then you might have little r, big Y, and that would leave you little r, little y. Yeah. You can have all of those. And that's where it relies on the independent assortment. All right. So those plants will produce 4 types of gamete. Those are all different gametes, different egg cells, different pollen cells in this case, because it's a plant. But you've got to have one copy of each gene, remember. And I think this is the one where people make the biggest mistakes. This is where I get, you know, big R, big Y, little y, big R, big Y, big R. I get 6 sometimes because I might get those 4, plus people will put the R's and the Y's on their own as well, which, you know, is ridiculous. But obviously for this one we get the same. So we've now got to think about, okay, combinations, or how we're going to work out the offspring. Can't do a crisscross with this because we've got 4 on each side, and we've got, what have I done? I've put 2 little y's, haven't I? After saying that I don't know how students can be so daft in exams, I've actually done it. Hey ho. There we go. Did anybody copy me? No, good. That's the trouble when I leave my hand to get on with writing while I'm talking. That's, yeah, important. So we've got to put these together now in all possible combinations, and this is where we can't do a crisscross for this, and this is where a square works. All right, now we call this a Punnett square, named after somebody called Punnett, I think. And you don't just have to do it with these where you've got 4 possible gametes from each parent. You can do it for smaller numbers, and that's what we did for the, you know, that was the other way of writing out the monohybrid one, is to use a little square for all the combinations. You are going to need a bit of space for this. What you need is, see if I can put it in as a table. I suppose it'd be 5 by 5 to get all the... I wonder where that went. I love my smartboard, honestly. I just need to move it. Come on. So there's a reason. You sort of have to... no, I can't now get the whole thing. Right, okay. Yeah. I think I needed to extend the page to put it on. No, we're all right. Okay, that should do the job. Yeah, it's rather curiously, you can't actually make it any bigger now, I don't think. Yeah, I can stretch the bottom down like that, but that doesn't really work. But I think you'll be all right like that. Okay, now, best way to do this. It doesn't really matter which goes where. I'm going to split that one. So we've got one parent going along the top, one coming down the side. So either way round, let's put the female parent along there and the male parent down the side. And I'm going to use a different colour just so that things stand up a little bit, stand out a bit better. All right, let's use the red. Okay, those are your possible gametes from the mother plant. Now, I could do the ones from the father plant in another colour, but that would then take me ages to write out the combination, so I'm not going to. This works better if I can get the actual rows a bit squarer than that. Let's see if I can stretch them a little bit, or quite a lot. Okay, so bear with me, it's going to be a little bit messy for a couple of minutes. And I have to move the... it's all right, I can do it. Ooh, that's come out big. Oh, wow. Ah, okay. Oh, I could just delete them and start again, couldn't I? Looks a bit silly. The reason I'm doing this is because I want to be able to draw the shape of the P. I just needed a bit more space. Okay, now if you haven't yet realised, all you've got to do is then put these together. So each of the remaining squares has got to contain 2 R's and 2 Y's as determined by the row and the column. So if we go across the top, big R, big R, big Y, big Y. Then you've got big R, big R, big Y, little y. You've got big R, big Y, big Y, big R, big Y, little y. And so you go on. So you should be able to do those yourself and then check. I'll just carry on and do them. I'm not going to try and read them out as I go, but I'll put them up so you can check when you've done. If you're not following how it works, for goodness sake, say so. Right. If you don't agree with what I've put, let me know, because if know I'm perfectly capable of making mistakes. Running my eyes over them, they appear to be okay, but you know, it's like proofreading your own work. You don't always see. That matches, yeah, okay. So once we've done this, we can then sort out the phenotypes. Now there isn't really room to write them in there, so I'm going to use well, a visual way of doing it. And I need, oh no, I've put that back. Right. Okay, so let's determine whether these are round, wrinkled, yellow, green, in what combinations. So what's this first one in the top? Round yellow. Next to that we have round yellow. Next to that, round yellow. And next to that, round yellow. Okay, and at the beginning of the next row we have, oh, I fancy that. Next one along, round wrinkled. Yeah. No, let's try that again. Round green, that's the word you're looking for. Next one. Yep. And yes. Okay. Next row. Yep. Oh. Yes. It's a bad one. Round yellow. Yay! We've got a different one. What's this one? Oh, how are we going to do this? Okay. Wrinkled yellow. And wrinkled yellow. Okay. Next row. Round yellow. Ah, right. I think that's the last of those, isn't it? Round green. Okay. Then wrinkled yellow. And wrinkled green down in the bottom corner. Only one way of getting those out of all that. Right. Okay. You know, you can use some sort of code. You know, if you need to describe what's what out of these, then you know, then entirely up to you. So how you do that. I should have said what I was going to do before I started, probably. Okay. How many different phenotypes are there? 4. Yes. All right. There's only 4 different physical appearances. Round yellow, wrinkled yellow, round green, wrinkled green. Yeah? All right. What's the ratio this time? You've got to do some counting up. Pardon? Are we getting there? I think I heard it. Yeah, we usually do it the other way round, start with the big one first. But yeah, 9, yeah, for the dominant. How many for dominant recessive? 3. How many for recessive dominant? And how many for recessive recessive? 1. So we actually have this ratio. It's known as the dihybrid ratio. which you only get when you are crossing two individuals who are both heterozygous for both characteristics. The dihybrid ratio is described as 9:3 to 3:1. Okay, now that's fine. If you're breeding organisms that produce quite a lot of offspring, then that's, you know, something that you can easily test, isn't it? Really, if you think about it, you know, you could, let's say, with think about typical lab animals, mice. You know, you can breed several generations of mice quite easily, or, you know, interbreed several pairs, and, you know, easily get perhaps 100 mice or something like that, couldn't you? You can do it with plants because they tend to produce quite a lot of seed. You know, in comparison with animals, plants produce a lot of seed because a lot of it's going to get eaten, isn't it? But of course, if you were looking at animals that don't produce so many offspring, it's a little bit difficult to test this. So if you were trying to test it with humans, for example, you know, you couldn't do it with one human pair. But of course, you know, just generally speaking, if you sample a population, then you should be able to find that sort of ratio in the characteristics, you know, between well, heterozygous parents, if you can find enough of them. But that's not easy in itself. So this idea, the idea of the dihybrid ratio, it is something that we can test theoretically using the chi-squared test. But I'm going to come back to that in a bit because I don't want to confuse matters too much. I do just want to sort of loop back to it. But, you know, if, for example, we had, well, what's our total number of offspring here? 16. So let's say you had 16 offspring. You might expect to find them in that ratio between those two parents. If you had 160, you might expect to find 90 to 30 to 30 to 10. But let's say you had 160 and you didn't find 90 to 30 to 30 to 10. I'll come back to this. Let's say you found, I don't know, 80, whoops, not 8, to 40 to 20. Oh, then I haven't got any left, have I? 40, that's 120. I've only got 20 left. Yeah, but I was going to lose one on the end. So let's say 15 to 5 or something like that. Could we accept that as being close enough to represent, you know, independently assorting characteristics? Is 80 to 40 to 15 to 5 close enough to 93 to 3 to 1 to accept? Yeah, I'm getting frowns and people aren't sure. Oh, sorry, have I added it up? Oh, sorry, yeah. Sorry, maths. That's why you were frowning. Ah, dear. 80 to 40 to... right. I've still got 40 left at this point, yes. Let's... Yeah, that was where I was supposed to be going, wasn't it? All right. Would that be close enough? You'd look at that and you'd say, yeah, probably. You know, that's not too bad, is it? But, you know, how far away would it need to get? Let's say 60 to... what if it was there? Have I got the right number there? 110, 50? Yes, I have, actually. Would you accept that one? Oh, well, that's the sort of thing you'd be looking for. It would be, yeah, are the two in the middle less than the two at the ends? Is the one at the left end the biggest and the one at the right end the smallest? Probably acceptable. But, you know, if it came out as 40 to 40 to 40 to 40, you'd probably say, hang on a minute, there's something strange happening there. So, you know, what is acceptable? What is an acceptable difference? And how do you decide that? And that's where the chi-squared test comes in, because with this ratio, we know what we expect to see. And the chi-squared test, and I can't remember when we did that, but it must have been about, I don't know, October, November time? Lifetime ago, isn't it? But at that point, we couldn't actually use it on anything, but because that's where I do the data handling bit, of course, you know, it had to be done there without any sort of examples. But what we'll do, we'll have a look at a couple of questions, if I've got them, on the dihybrid cross to start with, and then we'll come back to this, because I only need to run the, I think it's on a PowerPoint from that first unit. But this is the sort of thing that's actually used a lot in checking out genetic crosses. It does have a lot of other applications. It's not just used for this, but the chi-squared test tests observed against expected, so that you've got a way of saying, yes, this does support the hypothesis that these two genes are on separate chromosomes, or it doesn't support the hypothesis that the genes are on separate chromosomes. So it suggests that perhaps there's some linkage there. It might be that, you know, sometimes crossing over put them both together, or didn't put them both together. Let's say independent assortment then didn't separate them all out as you'd predict, and so the ratio, you know, is not exactly what you'd expect. Now, of course, you can actually go beyond the dihybrid cross, and I remember doing a genetics module at university in my third year, which admittedly is a long time ago, and we used to have to work out the trihybrid outcomes by hand. It took a while, but you know, for this you're just looking at the combinations of two genes. All right, so color, shape. If you were to add to that the height of the plant, big T for tall and little t for dwarf, then I can't remember how many combinations you get from that, but I could work it out quite quickly, I'm sure, if I, you know, felt I had to, which I don't at the moment. So I'm just pausing at that point for now, and we will have a look at, well, if we have a look in the handout folder, we are getting ever closer to the end, and we should find that there should be, I say there should be, I'm hoping there are, we've got the lethal, oh right, okay, I haven't put them in, all right, I haven't actually put them where I should have put them. I've put them in mixed with the population genetics bit, with what you can see is known as the Hardy-Weinberg. I appear to have, yeah, not put in any ordinary. No, I haven't put in any ordinary dihybrid crosses in there. Okay, we'll pass on that one for now then. That was something I should have checked beforehand, isn't it? Right. What I'm going to do then is have a bit of a, you know, return to the chi-squared bit that we did, the chi-squared test, which I'll try and find it. I know you won't have your stuff here. You're not cutting about the handouts for cells still. I wouldn't have thought. Hopefully you've still got them somewhere, but somewhere at the bottom of a pile, I would think, seeing as that was the first unit we did. I think that would have been the last one, wouldn't it? We did have some examples. I can't remember whether we, sort of how many we might have done, but the problem that we had when we were doing this was that it required a table of chi-squared values. All right. But we'll work through an example, and I'll just show you sort of how we could use this. Right, let's just run this through. It is worth hanging on to, notes from other units, because you never know quite where they're going to come up again. It's all very well to study a unit at a time, but it does risk sort of isolating the content and suggesting that there's no overlap with other areas. The cells one, I think, probably has more overlap than anything else, because all living things are made of cells. And this bit, of course, it does relate to all the sort of practical work and data handling that you might come across. We did, calculation. I think this year I sort of thought a little bit more about correlation than anything else. And there are various statistical tests you can use. I'm just trying to think what else you come across. You're going to use the Simpson's Diversity Index in environment, which is another way of looking at variation between populations. It's a matter of looking at the diversity of birds in one location, and then looking at the diversity of birds in another location. You don't get an answer that has any units or reads off a scale or anything like that. The purpose of that is to come up with a number for here, a number for there, a number for somewhere else, a number for another place, and say, right, well, a bigger number, greater diversity, and it's as simple as that. It is just a comparative measure, I think, as far as I remember. Oh, how do we do that? I wasn't expecting that to happen. Right. Oh, I think I clicked on something at the bottom. Yeah, there's some little icons down there. Now, where is it? Please let it be in here. Yeah, this was where we started looking at statistical testing. When you get to university, I would think almost regardless of the course that you go on, there will be some degree of testing results against what's expected. The more biological your course is, the more likely you are to look at statistical results. But when you read research papers, they will usually say, if you're looking at a method that's been used, and then very often it will say, I'll try to think how it's actually described. The significance was calculated using ANOVA, A-N-O-V-A, is a statistical test, and I can't remember what it stands for. I think it's variance analysis on the end. And you basically just put your numbers into a computer and it does it for you, which, that's okay, but you really don't get a feel for what you're actually doing. And you might think, that's fine with me, I don't want to work it all out. If a computer can do it for me, it's welcome to. And to be honest, yeah, with all that hassle, all the possible calculation for that correlation calculation, that was potentially quite huge. And just sticking it into Excel and getting the spreadsheet to actually do the working for us, yeah, much better. But you will see things like P equals 0.05 or that's what you'll usually see in research papers. This idea of statistical significance, because we tend to use the word significance a bit loosely, I think, in science, if we're not careful. It has got an actual mathematical, statistical meaning, the word significance. Are these results significantly different from what we expected? That's not just what's your opinion. All right, there is a number we can attach to it. So this idea of statistical significance is, if you look up there, the likelihood of getting a given result by chance. All right, so it's the probability of getting a result by chance. Now, biologically, obviously, chance in living systems is a huge player. So much happens just by chance in biology. So, there's something that it's quite useful to be able to put a number on. So, statisticians' criterion as far as significance is concerned then is the likelihood of getting a result by chance. Likelihood, if you say probability instead, that's where the P comes from. All right, so the p is the probability value, if you like, the p value. So the probability of getting a certain result just by chance, and we're happy with a p-value of 0.05. Okay, we're happy with it, being, as long as it's more than a 5% chance, 5% probability of getting it by chance, we're okay with that. Which actually seems quite low, doesn't it? You might say, well, so 10% probability that it's just from chance. You'd think, well, I'm not sure that's very good, but that's a very acceptable result. And you use this mainly on what we call a null hypothesis, which is there won't be any difference between test subjects and sort of like the placebo ones, the control group, if you like. And you can actually use much lower numbers than that as well, 0.01 or 0.001 if you're doing things like drugs trials. So 0.001 would be 0.1%, so 1 in 1,000. All right. Now, when you're testing the effects of drugs, then yeah, okay, we don't want that to be due to chance, do we? That somebody gets better or has a certain side effect. So having a 1 in 1,000 chance that happens, you can see now why you actually need quite small numbers, I hope. Right, let's see, did I? No, I didn't, did I? It's not on here. Oh, heavens. Right, sorry. Oh dear, I thought I'd actually got it on there. We must have worked from sheets then, I'm assuming. Right. Right, this is a slight aside, as it were. So it's not something that I'm going to ask in the exam, but it has a bearing on considering results, if you like. All right, so I wouldn't ask it directly. I'm not going to get you calculating chi-squared tests, but it's to make you think about whether the results are always just going to be what you predict, which they're not, of course. The chi-squared test, of course, chi, and it's a good job Alan's not here, it's a Greek letter. As is a lot of science. I mean, you practically do need to learn Greek for science, don't you? There's so many of the letters are used. It's an uppercase, I don't think they call it chi, it's more of a chi. It's one of those sounds that we don't use it in English, but it's one of those sounds. It's halfway between an X and a K sort of sound, something like that. We still say chi-squared and then we write chi. Yeah, if you're going to call it chi, why don't we just put Ki? Who knows? But the Greek letter is, known to us as chi. Or it might be chi, I have no idea. I get a bit obsessed by it sometimes. But this, as I say, is a bit of a review. It's almost sort of closing loops back to that first unit, because it's all very well to treat the course as a linear thing as well. Oh, we have to do this, then we do that, then we do that, and we're now a mile away from where we started. But actually, I do try and put in more than one sort of loop back to the beginning. It's like, doing respiration further down the line than when we did mitochondria. That sort of thing, trying to sort of pull it all back together again a bit. So this loops us back to the cells unit, but not so much cells, it's about the data handling, which could turn up anywhere. So what this does, it tests observed results against expected. And you end up, you get a value for chi-squared, by, I've now got to try and remember the formula, and you work out what we call the degrees of freedom. Does that ring a vague bell? The degrees of freedom is the number of categories of result you've got minus 1. And I cannot tell you why, because I used to find statistics hideously boring and I never listened. I've looked it up since, and you know how your brain just shuts off when you're reading things sometimes? I just don't care. And I think I really ought to, but I think it's just to do with allowing for the number of individuals in a sample. Because obviously, if you're comparing observed against expected and you've only got, let's say, 2 possible outcomes, you're going to get different results from, if you're comparing observed against expected, but there are, let's say, 20 possible outcomes. And allowing for that is where we include this idea of the degrees of freedom. It adds this contributing factor for that. So we get a result for chi-squared. Now I've got to try and remember. And we know how dangerous that is. So, and it's probably a better idea if I just have a look back. I could look it up, but I know that we had in here. Yeah, there were some sheets, weren't there? Yeah, so yeah, I had here we go. That's yeah. This was the I think I gave you these separately, didn't I? And yeah, for some reason I don't think they might be in the handout file. Yeah, I think they are, aren't they? So yeah, this, let's take that out of there and stick it in here. Save me trying to write it. Yeah, right. Get rid of that one. All right, there is chi-squared. So chi-squared is the sum of observed minus expected squared divided by expected. We then have to look up a value of chi-squared on a chi-squared table for the appropriate number of degrees of freedom. So, well, let's just do one. We've got time, actually. So, for example, if it would be quite nice to have, and I don't think I put any genetics ones in that one. I don't know if there were any that we couldn't do at the time because we hadn't done genetics. I think I actually told you that this ratio would apply. Oh no, everything's going to go wrong in the cupboard if I try and pull that lot out. Oh, right. All right, let's just take a random example. If in a plant, white flowers and dwarf height. That doesn't really make sense. Dwarf. It's not dwarfism in plants, but the trait is described. You have tall or dwarf. We talk about dwarf runner beans and things like that, rather than short runner beans. No, dwarf, not runner beans, French beans, I think. If those are, let's have those dominant. And red and tall are recessive. And across, this is getting a bit long, between two heterozygous parents gives, right, if we were to, I did scribble down some numbers from another example just in case I couldn't find the one I wanted. If we've got, for white flowered short plants, we ended up with 206. Red short, we had 83. For white, I should have put dwarf, not short, shouldn't I? Never mind. It's neither in or there. Tall, well, we got 65. And for red tall, we've got 30. Okay, if we get that, can we assume these genes are on separate chromosomes? Okay, this is just how we would use this. All right, so a reminder. Well, if they were on separate chromosomes, we'd have the ratio of 9 to 3 to 3 to 1. That's it. So from that, we can work out what our expected numbers would be. All right, those are our observed. All right, so, how am I going to do this? I need to make this easier, so I'm just going to put WS for white short, RS for red short, WT for white tall, RT for red tall. The observed is 206, 83, just about to write a 5, 65 and 30. So what are the expected results? How would we work them out? Add them up, yeah? Yeah, you got it. Add them up, divide by 16, and then do 9 to 3 to 3 to 1 on them. Yeah? So that lot added up is... Oh, this is going to take longer than I... It doesn't take long if you just get on with it, does it, rather than looking at it. All right, so we've got a total of 384. Okay, so whoops, what's happened there? Yeah, no, I just turned the calculator on, and rather than saying 0, it says 7. I have a feeling the battery's... Oh no, it's not got a battery. It's light one, but I think it's been covered up. That's interesting. Let's see. Right, let's have a look. That's better. Right, 384 divided by our 16. Yeah, 24. So we know that the expected number of what would be 24? The red, yeah, that one. That's a 1. All right. We can do the two middle ones in our heads because they should be 3 lots of 24, which is 72. Thank you. I thought the random number generator only went off in the other group. And that would be, what, we left with 9 lots of 24. I'm not sure I can do that in my head. Not reliably. 216. Okay, now looking at that, I wish they'd given us one that was actually, I wish I'd got one that was a bit further off, actually. You'd accept that anyway, wouldn't you? You know, that really isn't far off. But the way that we'd lay this out would be to calculate, and I don't think I'm going to do this actually because it's just going to be sort of long-winded. But you'd calculate observed minus expected for each. Then you do observed minus expected squared for each. I don't know if you can remember why we square it. That's right, gets rid of negative numbers. We then, if you look up here, have to divide by the expected. That brings it back down again. And then you can add those up. So it's then that okay, so you get your chi-squared. I've just sort of lost the will. I don't feel like calculating. Once you've got a chi-squared value, you then need a chi-squared table. And you had bits of chi-squared tables in your handout folders for this. And yeah, the trouble is every time I've finished a unit, I've just stuffed the folder back into the cupboard. So it's, yeah, a little bit messy in there. That's physiology, that's energetics, that's just can't be. No, that's biochem. No, I've got, it'll be right at the bottom of the heap, so. Once you've got a value for chi-squared, how many degrees of freedom? 3, yeah. So 4 results minus 1. So you look up on a chi-squared table to find probability of this, it's this deviation from the expected. Being due to chance. And I would think that's probably going to come out at least 0.9, I'd have thought. It's so close that the, yeah, I think the results are not going to be far from perfect. To find the probability of this deviation from your expected results. It doesn't say probability, it says probably. Yeah. Probability of. To find probability of this deviation from expected results being due to chance. When I do this, I always start saying, oh, to find the chance of this deviation from expected results being due to chance, and end up with chance in the sentence twice. To think very carefully and say, it is a probability, it's this p value that you get. It'll be between 0 and 1. 0 would be, this just didn't happen. 1 would be, if you got 206, 8, oh no, sorry, 216, 72, 72, 24. Well, there's no deviation actually. But the yeah, the probability of it being due to chance would be high because it's not much of a deviation. And the use of chi-squared in testing the outcomes of dihybrid crosses is it's very useful. If it's high, if you feel like calculating it out, you can do. So if P, well, it's only got to be greater than 0.05. the results are close enough to the expected ones not to be considered significantly different. And of course the question was, does this support the suggestion that the genes are on separate chromosomes? Yes, because you only get that 9:3 to 3:1 ratio if you are getting the full separation, the independent assortment. So close enough to expect it not to be considered significantly different. This would support genes being on separate chromosomes. I think I've written more now than I would have done if I'd actually gone through and worked it out, to be honest. But yeah, let's say you don't need to try and get your head around that for the exam on this, because it's not something I've written into any of the units, luckily. But it is something to be aware of. It is something that could come up, in future studies. Statistical tests are, well, they're a way of attaching a number to something that, very often is just due to chance, which is, I think, always a little bit strange. Just that because you've said it's unlikely doesn't mean it isn't going to happen. It's like the lottery. How unlikely is it now that somebody will win the lottery? How likely is it? They are not the same thing. But the probability of winning the lottery has dropped immensely, hasn't it? It used to be 1 in 14 million when there were only 49 numbers, and now it's, I can't remember how much how much more, or how smaller the odds are. But you'd think, why on earth does anybody now do the lottery? But every now and then I do it, because just because there is a huge likelihood of not winning doesn't take away the fact that there is a likelihood of winning. The one thing I have an argument with as far as the lottery is concerned is their advertising, where they use celebrities that they think people don't particularly like, and they say, please not him or please not her. Well, but that's not the point, is it? Because you playing won't stop them winning it. And it just is a really false way of trying to get you to play. You can't stop them winning. The best you could do was make them share. If you chose the same numbers and you both won, that'd be it. But it's sorry, you reckon? Well, there's but people still do win, don't they? So you've got statistics is a strange thing, but you can do whole degrees in statistics, and people work as statisticians, don't they, and are absolutely fascinated by the chance of things happening. And it's I think one of the biggest applications of probability is in insurance, because that's what insurance premiums are based on. The probability of somebody being likely to have an accident or taken ill on holiday, or their home burning down, or being flooded. And of course, if you've been flooded, if you're in an area that floods, the chances of getting insurance are not great, are they? Because the risk rises. It's that idea of risk with that. So really, all I wanted to do this for it was just to remind you about the idea of statistical testing and to reintroduce, that idea that you can attach numbers to things that otherwise you just end up expressing an opinion on. And just expressing an opinion is not actually very good. It's a bit like the error analysis, isn't it? When you have a go at that in chemistry, and I very rarely actually check out the actual numbers that people use in that. But if you've had a go, you get some credit for it. And that again is almost like sort of the probability of deviations in precision perhaps actually influencing your results. And it would always try to attach numbers to stuff. Okay, now the next thing that we're going to go on to, a little bit wary of getting too far in one go, and obviously we're missing a couple of people anyway, is population genetics. All right, but the other thing that I do want to do is to go back over the assignment, because I really want to make sure that people are now absolutely clear about what this entails. Okay, the assessment on this unit is an exam and the presentation. They are both worth 3 grades. All right, so there's all sorts of ways in which you could have an absolute wonderful outcome or a disaster. So please, I want to make sure you are exactly clear as to what you're doing. The assignment brief itself obviously it gives you the assessment criteria. Please don't forget the assessment criteria. All right, so it's got to be, you've got to have presented an account of an individually chosen and researched topic from the areas of applied genetics or recombinant DNA technology using PowerPoint or please not Prezi. I think I need to take that out. I don't want to encourage people to use Prezi. But if you don't know anything about it, sorry?
I've never heard of it.
You've never heard of it?
No, sorry.
I don't know whether I can show you very easily. Prezi, it was just something that for a couple of years I had students wanting to use it, but it backfired a bit because I assumed they knew how to use it and they didn't. They'd seen it and then sort of had to teach themselves. I had a go at teaching myself how to use it. But it's, you can try it for free. I have got an account on here, but I can't for the life of me think what it might, my login would be. Probably the college one. Goodness only knows what the password would be. Oh, no. Did it find me? Possibly. No, I don't want to do that. Oh, God. Don't want to do that. Just gonna... Why won't it let me? Can I get out of this? Is there a... No, there isn't an escape, is there? Yeah, it doesn't want to take... Oh, hang on. Aha! Right. Yes, look, here's one. Here's one I did earlier. This is one I did for GCSE biology a couple of years ago. I don't think I used it for you, did I? No. And it's I had to teach myself how to use it. Can't remember how to actually present it. So you see, it wasn't something, but you could expect to present remotely. I don't want to present it remotely at all. Yeah. Right. I think. Yeah. All right. Yeah. What it does, it allows you to do things like this. Yeah?
I mean, it looks fancy, but
It does.
yeah.
But you've got to learn how to use it, you can see, can't you?
Remember having handouts from that last year when it's kind of certain things on it?
Oh, what, from me?
Yeah.
Nah. You can't print it. Yeah, you can even put YouTube on it. Wow! Don't want to watch that. But yeah, it was a revision one for GCSE, and I put various things on it, and it was partly about me learning to use it as well. And then you have, and I don't know where this ends. Actually, it just seems to go on interminably. But you can sort of look at the whole thing, and I can't see quite where. I'm sure there's. I think under. Let's try the edit bit. I think you have a way of sort of looking at the whole thing. Just take a minute to actually come back up. But the idea is, and there's another one I've seen used within the last couple of years that's a similar sort of approach, but without the bubbles and things. You don't have to have circles on there. You can have all sorts of things. But the overall sort of presentation of this, you should be able to have a map of the whole thing, but I can't seem to see that anywhere. Okay. Nope. Okay. Must be a zoom out for it. Yeah. All right. So this is Prezi anyway. So if you were wondering, that's what it looks like, and you zoom into various bits of it. So unless you've got the time and the inclination, and you feel like learning something different, then I wouldn't bother, because it's not just setting it all up. You might see there's little numbers on here. You set up a path for going round it, so that every time you click, you go to a different part of it. I quite liked it, but it was awkward to learn to a new presentation software as well as actually putting the stuff on it. So I would stick with PowerPoint, but other presentation software is available. It's not. I'm not selling PowerPoint. The second of your assessment criteria: use appropriate illustrations to support the presentation. Now, appropriate illustrations, it doesn't mean any old illustration. It doesn't mean use lots that show the same thing over and over again. It means use illustrations that are actually going to help you explain something. They're going to illustrate something specific. They need a purpose, all right? Not just, say, a logo or a sort of GCSE bite-sized version of a genetic cross, or they need to have real relevance. Now, it'd be pretty hard, I must say, to actually fail on that. But you do also have grading descriptor 5 on here, which is about the presentation. So that will also be looked at in there. 6.3 is the hardest bit, I think, for a lot of people. Identify the significance of and/or ethical issues. So significance is about, if it's a genetic condition, how many people does it affect? What sort of influence does it have on their life? It could be about symptoms and treatment and that sort of thing. Statistics, what does it cost the NHS perhaps to look after people like this? Is it opening up areas of research? Is there therapy that's helping the development of knowledge in this subject? So it is that sort of thing. The ethical side of it, you may not have any ethics, but that could be about embryo testing or genetic counselling for parents, and the possible options that people have. But the ethics will vary from subject to subject, to be honest. So you do need to make sure you are going to tick the boxes on all of those. Your grading descriptors, number 4 is about use of information. are you using good sources? Now, this is not just about referencing. This is because that's in descriptor 5. Are you using sources which are reliable, suitable, all those sort of things? And are you using the information well? Okay, and then putting it together into a good account. Grading descriptor 5 is about things like your use of images, language, and referencing on the slides. So that's almost like, how good's your written work and diagrams on the slides. If you've got too much text, for example, or there are spelling mistakes, or your images are blurry, or you choose the wrong color combination, or your text is too small, that's where it's going to hit you. All right, so that's the one where you really need to make sure you've trialed it at least once. Well, I mean, James will have trialed it so many times. Just take up residence in a spare classroom, and yeah, that's all you need to do, because you'll find that the rooms along here become more and more available, and of course the more days that we're missing, you can still come and use boards and things. Although, of course, they will use some of the rooms, I suppose, for exams, won't they? That's the point. And then the quality one is the usual, does it flow properly and all that sort of thing. Now, alongside this, you must not forget that you have the study skills assessment, but just by turning up and doing the obvious things, you pass that, all right? It's not, I'd have to be really hard to fail somebody on that one. But the study skills one is to do with, we could do with looking actually, couldn't we? Because it's, where am I going to find that? Oh, it'll be in the URS, isn't it? Study skills, study skills, study skills. Can't see it. Must be easy. Oh, there it is. Yes, because it's a Carver unit. Right. Scroll down. No. It's stuck. Doesn't want to. Oh, here we go. Right. Let's enlarge that bit. It's learning outcome 4. I am going to assess you on 4 bits of that. You assess yourself on the 5th one. So 4.1, effective and appropriate communication skills for a target audience. All right. In other words, you've got fellow students who've been studying the same course as you. So it's a matter of using the right sort of language, being clear and loud enough, that sort of thing. But if you're not loud enough, we'll say so. I'll sit at the back and hide because I don't really want to be part of the audience. And if I can't hear you, I'll say, can you speak up a bit? I can't hear you. So that sort of thing. Or if you're going too fast or something like that, I will say so. So you'll get away with that one. Use visual aids effectively. Well, that's about, do you use your PowerPoint properly? Are you pointing to things? Are you using the right sort of images, the right sort of notes on there? I'm not sure what the difference is between 4.1 and 4.3 particularly. So if you pass one, you tend to pass the other one. 4.3, demonstrated interpersonal and behavioural skills by using appropriate style, pace and body language. So, yeah, that to me is effective and appropriate communication skills, to be honest. But, yeah, if you swear at your audience and hide under the desk, you may not pass that bit. All right. 4.4 is actually quite a useful one, because this sort of helps with the quality grade, actually. Used opening, signposting, summarising to structure the presentations. That idea of the logical flow and the fluency is about, setting the scene, just saying a little bit about why you've chosen that topic, not just going straight into it. Then having the sort of bulk of your content, and then just having the signposting really is about having almost, you could start, your first slide could be, although I know some of you are running up lots of slides already, you could have sections, but having, starting with an introduction that says, first of all, I'm going to talk about, where this where the gene is for this disorder, on which chromosome. Then I'll talk about its effect on the proteins, and then I'll go on to the symptoms that individuals experience, and then talk about treatments and research. Signposting, because you'll then have that on your slides, it'll be clear. And it is just about structure, that one. And then, so I will judge those, and you will do the last bit, evaluated own performance. Now, it matters not what you write in this section, because if you think it went badly, then you just write that. But it's about being honest and about actually being able to recognize the strengths and weaknesses, your good points and your bad points. If you knew your subject well, you can say, so, I felt I knew my subject well and, understood what I was talking about, but I went a little bit quickly on some of the slides and I noticed one or two misspellings, but overall I was able to answer a question that was asked, and if I was going to do it again, I might practice a little bit more, or, you just say those sort of things. And it is literally, have you been able to assess your own performance? It's not about your performance, it's about your ability to evaluate. Evaluate has got the word value in it. It means give value to things that happened. Evaluate is always, was this good? Was this bad? How good? How bad? That sort of thing. All right? So don't forget that's ongoing as well. Now, as far as the actual assignment, the biology bit is concerned, you've got, obviously, to produce a PowerPoint. Alongside that, you need notes to present from, which can either be at the bottom of the slides, printed off as notes sheets. Yeah, is everybody happy that they could do that if they need to? Or as a set of little cards, or just a sheet that you can work from if you like. Doesn't matter. And you'll hand those in. And no, I don't want a script. I don't want great long pages and pages of things you are going to just read. I would rather you didn't read. You will get better marks if you don't read, because it will show that you're using information better. But you may well you're needing to refer to your notes, because you're not going to have had that much time to study the subject. So you need a PowerPoint, you need presentation notes, you also need a one-sheet handout for your audience. Now, I used to say to people, oh, if you can't do, if you can't put one of those together, we'll just print off your presentation, in note form, like 6 little slides to a page. But that's a bit of a cop-out, really, because that's not, it's not that helpful to people. But it's quite nice, and this is why you did it in study skills as well, because it's part of that. And it's part of the communication, part of the grading, really. Are you communicating your subject well? And I would use the handout for things like maybe just a few bullet points, important things, or some things that you couldn't find room for on the presentation. Maybe a diagram, a flow chart, or some statistics or something like that, perhaps. Or you might have something on the presentation that doesn't show up clearly on a slide that you'd like people to have in front of them, and you could refer them to it. It could be a chromosome map showing where the gene is. That sort of thing. And your references. Okay? A reference list on your handout is really important because, you're sending your audience away having seen your presentation, and you would like them to remember it, and you would like people to think, oh, actually, yes, so-and-so did a presentation on that. I'd like to find out a bit more information. Oh, yeah, that reference looks useful. So, it's good practice to tell your audience where you got the information from, even though you are not necessarily going to be referencing on your slides. Some of you may. But I always say, it's fine just to do a references list at the end, because referencing on the slides can really get in the way sometimes. And you don't often see slides referenced. So, yeah. And so, well, citations on the slides, no, you see, I wouldn't worry about that. So, yeah. I'll leave you to be the judge of it, really, for images. Sometimes, they can be sort of not too intrusive, but sometimes they just seem to get in the way, particularly with an image where you've got a lot of words on the image anyway, and you really want people to focus on a diagram of something's happening here that I want to explain to you, reference or citation, down the bottom, and it actually just sort of detracts from what's on the, the information bit of it. Anything else I can think of on that? Not off the top of my head, so I think, any questions?